영어 발표를 준비했는데요. 이 한글 자막도 있으니 참고해주세요. Good afternoon, everyone. We are Gumi여고 High School students, and our name is 보라채, 하영안, 현아지, and 지용 Kim. Our presentation title is Assay of Biomaterial using enzyme coupling with supermolecule. We have carried out this project in Kumho National Institute of Technology. As you can see this slide, the risk factors of cardiovascular disease are many things. We have tried to use whole system concentration in fluid work. So, what is whole system? It was reported that the high whole system concentration in fluid caused the destruction of fluid world. Therefore, assay of whole system is very important. However, chemical structure of whole system is very similar with system. These two amino acids are different only in one methylene group. In this study, we focus on the new developed assay method of whole system using enzyme coupling with supermolecule. Before we go in, let me introduce the normal assay method of whole system assay. First, chemical method use the specific reagent which can bind with whole system, and immunological method use the antibody, and enzymatic method use enzymes that can catalyze with whole system. We use enzyme. We use enzyme which can bind with whole system coupling with supermolecule. Then, what is supermolecule? Supermolecule is relatively high molecular group having binding molecular forces such as hydrogen bond, electrostatic interaction, van der Waals force. Supermolecular chemistry can be used for molecular recognition and self-assembly.
We use supermolecule called Q-coupled, consisting of glycolyl lipid unit. Q-coupled have hydrophobic and hydrophilic property inside the cavity. Q-coupled 7 means it has 7 monomers. This slide indicates the specific binding of Q-coupled with homocysteine, not cysteine. The difference of hydrophobicity between homocysteine and cysteine enables this specific binding property. This slide exhibits total reaction pathway from methionine to cysteine. Two enzymes, cystathionin beta-6 and cystathionin gamma lysis, can use homocysteine as a substrate. These two enzymes produce hydrogen sulfide from homocysteine. So we can use these two enzymes for the assay of homocysteine. Now here we present the data from cystathionin gamma lysis. This slide indicated the circular structure DNA, which is called plasmid. The total size of plasmid is about 6.5 kilobase. Too much cystathionin gamma lysis genes exist here. And this plasmid was put into E. coli to produce HCGL. This process is called transformation. we purified plasmid DNA from transformed E. coli. This slide showed the DNA agarose gel electrophoresis data. We can see the 6.5 kilobase band by one side restriction enzyme cutting sample and 3 bands of non-coated plasmid. These 3 bands indicated the supercoiled plasmid, linear plasmid, and circular plasmid. From this data, transformation process was good.
And then we check the protein production. We use polyacrylamide gel electrophoresis. Method 1 is SDS phase, and we can check many HCG proteins that normally collide at the size of 45 kilodalton. In native phase, we can check protein activity. And black color band means the hydrogen sulfide produced by the enzyme. From SDS and native data, we can suggest HCGL was produced in transformed E. coli. And then we check the HCGL using Western blotting, which is immunological assay. E. coli having plasmid was stained by antibody of HCGL and CBB. These two data means that HCGL was produced successfully. Using affinity column chromatography, we could purify HCGL. We can check only one protein band at SDS phase of about 45 kilodalton. Here is the experiment outline. Human cystathionine gamma lyase can produce hydrogen sulfide from cysteine. If we premix Q-Coptril with cysteine, Q-Coptril cannot bind with cysteine, and cystathionine gamma lyase can produce hydrogen sulfide, and we can see black color will be formed in lead acetate solution. However, if we premix Q-Coptril with homocysteine, Q-Coptril can bind with homocysteine, and cystathionine gamma lyase cannot produce hydrogen sulfide, and black color will not be formed. Now we can design the experiment how we can assay homocysteine in the presence of cysteine. If we can calculate the binding affinity in the presence of Q-Coptril, we can assay the homocysteine concentration without interruption of cysteine.
As we expected, pre-incubation Q-Coptril with cysteine gave no observance change, but with homocysteine shows the observance decreasing depend on Q-Coptril concentration. We suggest the decreased value indicated homocysteine concentration in the sample. Oh, no. Here the decreasing was stopped because homocysteine amount was limited. From the data, we could calculate binding constant between homocysteine and Q-Coptril using kaleidogram. We used this equation. Binding constant K value means the half of homocysteine concentration indicates the affinity behavior of two materials. As a result, we obtained the result 26.7 micromole in Q-Coptril homocysteine complex. It means we could assay homocysteine in micromole levels. This is conclusions. We could homocysteine and cysteine by using HCGL, and we have developed the homocysteine using enzyme coupling with supramolecule for the first time